For most of human history, aging looked like an obvious fact of life, not a scientific puzzle. Things wear out, bodies break down. That seems self-evident enough that nobody thought to ask why it happens the way it does. But aging is stranger than it looks once you actually examine it as a biologist would. Some animals barely age at all. Certain jellyfish can revert to an earlier stage of their life cycle and start over. Naked mole rats show almost no increase in mortality risk as they get older, for the majority of their unusually long lives. Meanwhile, humans, and most mammals, follow a predictable trajectory of decline that seems locked in from early adulthood onward. If aging were simply an unavoidable consequence of physics or chemistry, wearing out the way a machine wears out, you would expect it to look roughly similar across species with similar cellular machinery. It does not. This inconsistency is the first clue that aging is not a fixed law of biology. It is something evolution shaped, and shaped differently in different lineages. Which means the real question is not just what happens when we age, it is why evolution allowed aging to happen at all, given that natural selection usually favors traits that help an organism survive. That question has produced several major theories, and they disagree with each other in genuinely interesting ways. The first serious evolutionary theory of aging came from a British biologist named Peter Medawar in 1952. Medawar's insight was simple but reframed the entire problem. He pointed out that natural selection's power to weed out harmful mutations depends entirely on when those mutations cause damage. Imagine a genetic mutation that causes a fatal heart condition at age 12, before reproduction. That mutation is under intense selective pressure. Anyone who carries it is unlikely to pass it on. So the mutation tends to get removed from the population over generations. Now imagine a different mutation that causes the same fatal heart condition, but only at age 70, long after most reproduction has already happened. Natural selection barely notices this mutation. The person carrying it has likely already had children and passed on their genes before the mutation does any damage. There's little to no selective pressure removing it from the population. Medawar called this the selection shadow. Beyond the age when reproduction has typically finished, natural selection's ability to filter out harmful mutations drops off sharply. Harmful mutations that only manifest late in life can accumulate in a population over evolutionary time, simply because selection cannot see them well enough to remove them. This theory explains something important. It explains why aging exists as a general phenomenon, but it does not fully explain why aging looks the way it does, with specific pattern types of decline showing up across many species in similar ways. For that, a related but distinct theory picks up where Medawar left off. In 1957, an evolutionary biologist named George Williams proposed an idea called antagonistic pleiotropy. Pleiotropy refers to a single gene affecting multiple traits. Williams's idea was that some genes might be actively beneficial early in life while being harmful later in life, and that natural selection would favor these genes anyway, because the early benefit matters more for reproductive success than the late cost. A commonly cited example involves calcium regulation. Genes that promote strong bone density and efficient calcium absorption in youth may be advantageous for growth, strength, and fertility. Those same genetic mechanisms operating over a much longer timescale may contribute to arterial calcification and cardiovascular problems later in life. The gene is not simply neutral early and harmful late. It is actively helpful early and actively harmful late, and evolution takes the trade. This is a genuinely different claim than Medawar's shadow of selection. Medawar's idea says evolution fails to remove late life damage because it cannot see it. Williams's idea says evolution will actively select for late life damage if it comes attached to an early life benefit, because early benefits count for more in reproductive terms than late costs do. Put together, these two ideas form what is often called the evolutionary theory of aging, and for decades it has been the dominant framework in the field. Aging, on this view, is not a program the body runs. It is closer to an unintended byproduct of a body optimized overwhelmingly for early survival and reproduction, with the cost of that optimization deferred to later in life, where selection has less power to intervene. There is a third theory worth understanding called the disposable soma theory, developed by biologist Thomas Kirkwood in the late 1970s. This one focuses less on gene selection and more on resource allocation. An organism has a limited amount of energy available. That energy has to be divided between two competing demands: maintaining and repairing the body and investing in reproduction. Perfect cellular maintenance and repair, the kind that could theoretically prevent aging altogether, would require an enormous amount of energy. Kirkwood's argument is that evolution does not favor organisms that invest heavily in perfect bodily maintenance. It favors organisms that invest just enough in maintenance to survive long enough to reproduce successfully, and diverts the rest of that energy budget toward reproduction itself. The name disposable soma comes from a distinction in biology between the soma, meaning the body, and the germline, meaning the reproductive cells that carry genetic material into the next generation. Kirkwood's framing is almost unsettling when you sit with it. From evolution's perspective, the germline is what matters. The soma, the body carrying it, is somewhat expendable once it has done its reproductive job. It only needs to last long enough. This theory helps explain something that Medawar and Williams's ideas alone do not fully account for: why species with different reproductive strategies age at such different rates. Animals that face high extrinsic mortality risks in the wild, meaning they are likely to die from predation, starvation, or environmental danger regardless of internal aging, tend to evolve fast reproduction and fast aging. There is no evolutionary payoff to investing heavily in long-term bodily maintenance if you're statistically unlikely to survive long enough to benefit from it anyway. Conversely, animals with fewer natural predators and safer environments tend to evolve slower aging and greater investment in long-term repair. This is part of why naked mole rats, who live in predator-protected underground colonies, show such minimal aging compared to similarly sized rodents living in more dangerous environments above ground. It is also part of the explanation for why larger animals with fewer natural predators like elephants and whales tend to live dramatically longer than smaller prey species. These three theories, the selection shadow, antagonistic pleiotropy, and disposable soma, are not competing explanations so much as complementary layers of the same underlying picture. Aging happens because natural selection's influence weakens with age, because some genes trade early benefits for late costs, and because organisms are shaped to prioritize reproduction over indefinite bodily maintenance. None of these forces requires aging to be programmed or intentional. They only require that evolution optimizes for reproductive success, and that reproductive success mostly depends on what happens early in life. This raises an obvious and increasingly relevant question. If aging is not a fixed law of physics but a byproduct of evolutionary trade-offs, could it in principle be slowed or even substantially reversed through deliberate intervention? This is where the biology gets more speculative and more actively researched. Several biological mechanisms have been identified as central drivers of the aging process at a cellular level, and each has become a target for scientific intervention. One mechanism involves telomeres, the protective caps on the ends of chromosomes that shorten slightly every time a cell divides. Once telomeres shorten past a certain point, cells generally stop dividing or self-destruct. This has led some researchers to investigate telomerase, an enzyme that can rebuild telomeres, as a potential anti-aging tool. The complication is that cancer cells frequently exploit telomerase to divide indefinitely, which means artificially extending telomeres carries a real risk of promoting tumor growth. The same mechanism that could theoretically extend healthy lifespan is closely linked to one of the most dangerous failure modes of unchecked cellular division. Another major area of research involves cellular senescence. As cells age, some enter a state called senescence where they stop dividing but do not die. These senescent cells accumulate in tissues over time and secrete inflammatory signaling molecules that can damage surrounding healthy cells, a phenomenon researchers sometimes describe as cellular zombies causing collateral damage to neighboring tissue. A class of drugs called senolytics is currently being studied for their ability to selectively clear senescent cells from the body. Early animal studies have shown promising results, including improved physical function, and in some cases extended lifespan in mice, though translating these findings safely and effectively to humans remains an open and active challenge. A third avenue focuses on caloric restriction and the cellular pathways it activates. Studies across many species, including yeast, worms, flies, and mice, have consistently shown that reducing caloric intake without causing malnutrition can extend lifespan, sometimes substantially. The leading explanation involves a cellular pathway called mTOR, which regulates growth and metabolism. Caloric restriction appears to shift cellular priorities away from growth and toward maintenance and repair, echoing Kirkwood's disposable soma framework almost directly. This has led to interest in drugs like rapamycin, which inhibit the mTOR pathway and have extended lifespan in laboratory animals, raising the question of whether similar effects could eventually be achieved safely in humans without the difficulty of maintaining long-term caloric restriction. None of these interventions have yet been proven to meaningfully extend maximum human lifespan, as opposed to average lifespan, which has already increased significantly over the past century, mostly due to improvements in sanitation, medicine, and reduced childhood mortality, rather than any change in the fundamental biological aging process itself. The distinction matters. Living longer on average because fewer people die young is different from actually slowing the intrinsic rate of biological aging in individuals who do reach old age. What the evolutionary theories of aging offer beyond their explanatory power is a reframing of what would actually need to be true for radical life extension to work. If aging exists because evolution simply never had reason to optimize the body for a longer functional lifespan, rather than because indefinite biological maintenance is somehow physically impossible, then in principle there may be no fundamental law of nature preventing significantly longer healthy lifespans. The obstacle would not be that repair beyond a certain point is impossible. It would be that evolution never selected for organisms capable of doing it, because there was no reproductive payoff for maintaining a body longer than necessary to pass on genes. That reframing does not mean radical life extension is close or even guaranteed to be achievable through current research directions. Biological systems are deeply interconnected, and interventions that alter one pathway often carry unintended consequences elsewhere, as the telomerase and cancer risk trade-off illustrates clearly. But it does mean the question of why we age has shifted over the past several decades, from a question with an assumed and unchangeable answer to a genuinely open area of active scientific investigation. We age because natural selection stopped watching closely after we finished reproducing, because some of the same genes that make us thrive young quietly work against us later, and because our bodies were built to prioritize passing on genetic material over indefinitely maintaining themselves. None of that was inevitable in any deep physical sense. It was simply what worked. over millions of years for passing genes to the next generation. Whether that trade-off can eventually be renegotiated is one of the most active open questions in modern biology.